Good evening, everyone. I'm Dr. Mikki McComb-Kobza. I'm the director of Ocean First Institute, and I'm so happy to be here tonight. I want to thank Sarah very much for the introduction to Aaliyah, the president of Finstitute. I'm so excited to be here tonight to talk to you about hammerhead sharks and my research. So I'm going to go ahead and share my screen and get started. All right, perfect. Okay. All right, so hammerhead sharks, I'm going to talk about them tonight. And so shark skates and rays, as you know, have been on the planet for over 400 million years. They are older than the dinosaurs and the trees. They are collectively known as the elasmobranch. They are the cartilaginous fishes. And because they've been on the planet so long, they have radiated into nearly every aquatic habitat on the planet. And we see marvelous adaptations in these animals. Over 500 different shark species are extant and swimming in the ocean today. And again, all have different adaptations, the way they reproduce, the way they swim, everything about them is unique in the adaptations to the environments in which they're found. I have been a shark researcher for over 20 years, and much of the research that I have done has focused on sensory biology, all the way to movement ecology and conservation. And I am I'm faculty at CU Boulder in the Ecology and Evolutionary Biology Department. I'm the president of the American Elasmobranch Society. I'm also a national fellow with the Explorers Club. And I also am the executive director of Ocean First Institute. That is a nonprofit organization, and our mission is ocean conservation through research and education. And so tonight I'll be talking to you about the research that I've done on hammerhead sharks. Hammerhead sharks are unique in that they are the most recent evolved sharks in the ocean. Very new to around 10 to 12 million years ago, we started to see hammerheads. And what you might not know about them is that there are multiple hammerhead species, all with different gradations of head expansion. And so when you look at a hammerhead, you have to think why? Why do they have the weird head shape? What is the benefit? Is it adaptive or maladaptive? And what's interesting is in the scientific literature, there have been arguments and heated debates about why hammerhead sharks have the weird head shape. And so when you look at the different species here, you can see that there are ones that look almost like normal sharks all the way down to what looks like a boomerang head and everything in between. And so the hammerhead that looks like a boomerang is in its own genus. It's called Eusphyroblachii. And what's interesting is that the genetics, the phylogeny of hammerheads puts this as basal, so the very first hammerhead, and puts the bonnet head as the most derived, the most advanced. And so it's a really interesting thing to think about what have been the pressures to drive the evolution of the head shape. And again, what's the functional significance of the hammerhead head? And so this is what the wing head looks like. I was able to spend time with this shark in Australia. This is a shark that has a very limited distribution. Most of the hammerheads, like the scalloped and great hammerhead, are found, they have large ranges. This shark does not. It's also very small. It only is a few meters long, and its total head width is about 50% of its total body length. So it's a very interesting looking boomerang-like shark. And if you look at an X-ray of a winghead, it really brings home, I think, the expansion of what's called the chondrocranium, and all of the sensory structures inside of that head have really been moved. And the question, can be what has happened to that sensory system in the sharks. And that's something that I focused in on. But again, there have been many different hypotheses that have been put forward to explain the function of this bizarre head shape. And one of those is that it serves a hydrodynamic function. So this is a Canard aircraft, and similar, you see wings in the front and back. That's what we see in hammerheads. And it truly is supported in research that there is a hydrodynamic lift by having this head shape, as opposed to normal pointed nose sharks. They're highly maneuverable. So we see that these sharks can truly turn on a dime. Some of the species of hammerheads use their heads to pin down their favorite prey, which includes rays. Not all of them do that, though, because not all of them have heads large enough to do that. And then, of course, there's the sensory function. So how does the expansion of the head, does it have a functional significance to the shark? Is it one of the evolutionary drivers of this large head shape? That was a question I asked for my PhD. And it was a very interesting question on many different levels. And one of the first ones to examine was vision. So, vision in sharks, is kind of surprising. It's their eyes are as complex as our eyes. They have mobile pupils, so they can dilate their eyes to control the amount of light coming in. I did a lot of experiments to look at what colors sharks could see, what their spectral sensitivity is, how well can they see in low light conditions and things like that. But tonight I want to talk a little bit about just visual fields. And so visual fields are the most fundamental aspect of vision. It's what an animal sees around its head. And what we typically see in predatory animals is that they have large binocular overlaps and frontally positioned eyes. And in animals that are more prey-based animals, ones that are being visually vigilant against predators, they typically have laterally positioned eyes that allows them and affords them large visual fields all the way around the head. And so I was interested in looking at the visual fields of hammerheads because my thought was they probably can't even see straight ahead with their eyes being pointed straight out on the ends of their hammers. And so I wanted to look at hammerhead visual fields in comparison to other normal head-shaped sharks. And what I found, the technique that I used is called an electroretinogram. So I had to go into the field, capture multiple hammerhead species, multiple individuals, and then bring them back into the laboratory where we are able to do basic visual testing. So sharks can't talk, and so I had to do the tests using a technique called an electroretinogram. So eyes are electrical organs, and as light impinges onto the eye, it sends an electrical signal down the optic nerve to the brain for processing as vision. So if you were to place a wire on the eye, an electrode, and shine light onto that eye, you can pick up the electrical signal that the eye generates. And that's what an electroretinogram does. And so in these experimental tanks, I would catch sharks, bring them back to the tanks, ventilate them. I anesthetize them. So if you were to go into surgery, you would have anesthetic. That's what we did for the sharks. So they go to sleep. And then we were able to put lights all the way around different parts around their heads to determine their three-dimensional visual field. So we would shine light on, see if we got an ERG response dictating that they're seeing, and we could figure out their three-dimensional visual field. And so what we saw was really interesting. It was a surprise. So normal sharks have about a 10-degree binocular overlap. And again, it's believed that binocular overlaps and binocular vision evolved to facilitate depth perception, which is important if you live in a nearly dimensionless ocean space. You need to be able to judge distances. And so it's important. And what we found, though, that was so surprising is that the hammerheads have anywhere in the bonnet heads to about 13-degree overlaps, to scalloped hammerheads 30 degrees plus, and then in the winghead almost 50 degrees. So a big surprise. And again, it was all based on the positioning of the eyes canted on the ends of the hammers. One of the other things, however, that I discovered in doing this visual field research is that there were large blind spots just anterior to the head. So you can see the shaded green area there is the binocular overlap, but we determined large blind spots. So just as you put your finger on the in between your eyes, you can't really see. Well, these blind areas went out almost a meter in length in some of these sharks. And so the question is, how do they see? What do they do if they can't see their food? So again, blind areas, large, and it got me to start to think about other sensory systems. So as you're sitting in the audience right now, you're using your eyes, you're listening, you're feeling temperature, you're doing all these things in combination, and sharks are no different. And so their eyes are paramount to seeing, but if they can't see in that visual void, what other things can they be doing? Well, of course, the sense of smell plays a part, but there's also the shark's very secret sense, the electrosense. So this beautiful image of a bonnet head on the underside, you can see these beautiful patterns of these peppery dots. That's the ampullae of Lorenzini. These are jelly-filled canals within the head of the shark, and they allow the shark to actually detect weak electric signals given off by living things. Every living thing in the ocean gives off an electrical signal, and sharks evolved to detect it. And so that electrosense is unique in each species. It has a different pattern, which correlates to their ecology, which is absolutely incredible. But I wanted to look at that interplay between vision and the electrosensory system. And so the way that we did this is my advisor, Dr. Steve Kajura, had been looking at the electrosensory system for years. And what we did is we mimicked Cray voltages, so around 6 microvolts, by creating dipoles underwater with batteries. And the sharks would literally come in when we flipped the switch and bite at this weak electric field, this dipole we were creating underwater. So there's nothing there except our battery, and the sharks were coming in. And so this was a really neat experiment for us to be able to understand, the sharks were coming in. But then we wanted to quantify this. And so at Mote Marine Lab, I was able to work on these black nose sharks. And you can see this black nose comes in and then orients its body to the weak electric field that I'm generating with a battery. And that actual distance at which it's orienting is its detection distance. And so I was able to run these experiments on all different kinds of species of sharks in great numbers to try to determine how far out do they detect these weak electric fields. And what we found when we mapped it is that indeed, yes, sharks have the ability to have that electrosensory system extend into the visual void. And that's not really a big surprise, because if you think about it, these sharks have been around for millions of years. And so they're constantly fine-tuning their sensory systems to really be as adaptive as possible for that. And so the question is, was this one of the drivers of, the pressure for the head shape in hammerheads? And the answer is possibly. Having that additional ability to have overlaps, binocular overlaps, to have the electrosensory system, expand out into this head that's, over a meter long. Certainly, we think that they're maybe not any more, they're not better at detecting electric fields, but they just have this big sheer head. The width of their head is so much larger than a normal shark. So the detection ability is probably greater. So really, it's anyone's guess, but we think that there are definitely things that drove the adaptability of the head shape and what it meant for these animals. The only maladaptive thing that I've seen in studying hammerheads is their fragility on the long lines. They are ram ventilators. They have to continually swim to survive. And when they get hooked on the lines, they just don't make it. They're very fragile. And I think that has a lot to do with their small mouths that don't open very wide. And again, they have to continually swim. So they are susceptible to dying on long lines. And this is particularly sad because, in the years that I've been studying these animals, we've seen the global decline in sharks, and it has been driven by the need and the desire for fins. So shark fins, as you all know, are cut off of the sharks when they're alive. Which is really pretty barbaric, and they're dumped back into the ocean to die. And the fin trade gets anywhere from 40 to 100 million sharks per year killed. And it's really an unregulated and poorly reported trade. And unfortunately, it has led to the great hammerhead and the scalloped hammerhead becoming critically endangered species. So that is why it's really important, I think, for this work to continue on. Much of the work on electroreception has, been now working on trying to keep sharks off long lines by using different technologies to repulse the sharks using electric signals. And we're seeing some limited success in that, and trying to do widespread adoptions of that into the fisheries is kind of the next step. So there definitely are ways that this work has, been translated into conservation initiatives. And for that, I'm very grateful. And, I guess at the end of the day, what I'd like to say to all of you is that, if you're interested in doing shark research, please do. Please know that this field needs biologists. We need researchers to go into this field because these animals are teetering. Many of them are critically endangered. And to be an ally for the sharks and to spread their story to the general public, to overcome the fear that people have of sharks, is critically important. So I'm personally grateful for your interest in sharks, and I certainly am excited to get to know some of you as you move into the shark conservation field. So thank you all for your time tonight. I hope you have enjoyed this, and if you have any questions for me, please know you can always email me. So thank you all so much. Have a great night. Bye-bye.